All right. So like I said, this is the last journal for this 6 weeks. So that journal test will be on Monday. And then we'll kind of just be doing different things to review for your semester test, which will be over quite a few different units. And so we'll be working on that on Monday or Tuesday whenever we have extra time. So does anybody know the difference between regular diffusion and facilitated diffusion? Nobody? Okay, well, why don't you pull out your notes and see if you can answer it. You can almost just look at the picture. Top one is regular diffusion, bottom one is facilitated diffusion. No, that's osmosis is water. So any kind of diffusion is any kind like molecules are long and they're going from from high concentration to low concentration. But what does your picture show on your facilitated diffusion? What do we have? What is stuck there in the middle of that? That we talked about on the other page. If you flip back on the other side. Yeah, it's either it could be some kind of channel protein. It could be, like you said, it could be for ions, it could be for molecules, but facilitated diffusion has to have some type of channel protein. So that's going to be the difference between the two. Both are still passive, and both still go from high concentration to low concentration. But facilitated diffusion would have to have some type of channel protein. All right. So we're going to just kind of finish up the different types of passive diffusion today. Not passive diffusion, passive transport. So we're going to start with a little video here. Is that too loud? Is that too loud for y'all?
Diffusion is used as a transport mechanism in the cell.
Oh, that's not.
Small molecules can move from high concentration to low concentration by normal diffusion across the cell membrane. Large molecules cannot diffuse through the membrane easily. Thus, the cell uses a protein to aid diffusion.
All right.
Imagine the places we'll go together. Expedia.
What makes a business a business? It's who starts it.
Yes, sir.
I will never forget the first time my fish caught teeth. Well, it's called ick santos.
If you finish that,
It's actually caused by a protist, a parasitic one. In our ecology video, we talk about parasites.
Don't like this either. What happened? What am I doing?
Causing harm to their hosts. And in my case, it was
Oh no, this is still
beloved
diffusion.
fish. Well, okay, technically it affected birds and first, but then it spread because it is a contagious disease in fish. It typically causes these white dots. It gets on fins, on their bodies, on their gills, all over. It can even be lethal if not treated. So it's bad news, and being a guppy enthusiast, I was really worried about all of my guppies. This is not a video about parasites. This is a video about diffusion. So you're probably wondering, how is she going to connect diffusion to this parasitic disease? Well, it's because of the treatment. You see, there is a commonly purchased treatment for the disease of ick. One common treatment option contains an anti-parasitic called methylene blue. Its name is not misleading. It is quite blue. The directions on the back of the medication outline how many drops to add per gallons of water. And I watched and tried to encourage my fish to swim towards the medicine. But they were panicked, maybe because a portion of their water was changing colors. Also probably because of the kid trying to get their attention to swim towards the medicine. But you know what I didn't know then? I didn't need them to swim over there. The blue drops would spread on their own in a process known as diffusion. Diffusion is when the net movement of a substance travels down its concentration gradient. That means it moves from a high concentration to a low concentration. When the methylene blue drops were added into water, they didn't just sit there. The molecules traveled in the water through diffusion.
Okay,
High
so just
concentration
even finish that paper.
which is where they were dropped, spread to areas of low concentration in the
Now, what's
water.
up? They're gonna
Pretty
come get you?
soon, the water had a uniformly blue appearance. The molecules eventually are evenly dispersed. This was very important for treating my fish because the treatment reached all of them, and they soon got well. Diffusion doesn't just happen in water. For example, it occurs if you spray an air freshener in the air. The net movement of molecules travels from an area of high concentration of the air freshener to a low concentration of the air freshener. Pretty soon, it's likely others would smell the air freshener, even if they were quite a distance away. Now, I want to mention two important facts. First, we say net movement because that's the overall movement. But it doesn't mean that the molecules can't move around the other direction, and it doesn't mean that the molecules eventually stop moving either. The molecules are continuously moving, even when equilibrium is reached. It's just the overall movement, the net movement, is from a high concentration to a low concentration until equilibrium is reached. Second, diffusion is a passive transport. That means an input of energy is not needed. We say that passive transport does not require added energy. When we talk about diffusion of molecules in cells, for example, the diffusion of oxygen molecules into a cell, it is classified as passive transport. A concentration gradient is itself a form of potential energy. Passive transport is different from other processes that we detail in our cell transport video, like active transport, where an input of energy is required. And speaking of our cell transport video, we outlined that there is also a type of diffusion called facilitated diffusion. Facilitated diffusion is when molecules still have a net movement of a high concentration to a low concentration, but they may be too large or have other characteristics that prevent them from directly traveling across the selected cell membrane. Therefore, they have to go through a protein channel. It's still diffusion because it's still passive transport and it's still traveling down its concentration gradient. It's just that in that case, it generally has to go through a protein to get inside. Now, there are factors that can affect the rate of diffusion. We'll mention just a few of them when considering these small molecules here, diffusing in simple diffusion. Distance. The greater the distance that needs to be traveled, the slower the diffusion rate. For example, you could observe a difference in a 5-gallon tank versus a 55-gallon one. Temperature. Okay, really quick, would you think a higher temperature or a lower temperature would increase the diffusion rate? Assuming everything else is constant for the point of the comparison. Generally, a higher temperature. A higher temperature generally would mean there is more movement of the molecules, so the diffusion rate is increased. This has to do with energy, so check out the link in the description for more about that. Characteristics of the solvent. For example, is the solvent very dense? That could slow the molecules down and decrease the diffusion rate. Characteristics of the molecules, or whatever substance is traveling through diffusion, since technically not everything that travels in diffusion is defined as a molecule. What is the mass of the substance traveling? Generally, a substance with greater mass will have a lower diffusion rate when compared to a substance with less mass. Characteristics of the barrier, if diffusion is crossing a type of barrier. To explain that, let's consider diffusion across the cell, where the barrier would be the cell membrane. Small, non-polar substances pass through a cell membrane easier than something large or polar. That will affect the rate of diffusion. The surface area and thickness of the cell membrane also will alter the diffusion rate. For example, a large surface area and thin membrane would generally allow a faster diffusion rate than a smaller surface area and thick membrane. By the way, there are more factors that can affect the diffusion rate than just those 5 we listed. In fact, you may have already thought of another factor when seeing our simple diffusion image. Generally, if I were to increase the concentration of the substance here, that would cause a larger difference between these concentrations. and that would also increase the rate of diffusion. Before we end, you might wonder, why should I care about this diffusion rate? Other than the aquarium fish that I love, realize that diffusion is critical for all of life. Cells are the living units that builds up all life, and materials that cells need to survive have to get in, and waste molecules of the cell have to get out. Now, while diffusion is certainly not the only kind of cell transport, diffusion is responsible for many critical items. One example is you sit here right now, diffusion is responsible for oxygen, leading the alveoli of your lungs to your blood. Likewise, the waste gas carbon dioxide is able to exit your blood to the alveoli. Critical process made possible by diffusion. Well, that's it for the Viva Sisters, and we want you to stay curious.
All right, so that's just kind of a little bit of what we've been talking about. So to kind of finish up, passive passive transport, because that's all we're going to get to before our 6 weeks test. You have your worksheet that you'll use your notes on. But a couple of these do have a little bit about active transport. So I want to give you those before you get started. So let's see. On that top section, all right here, endocytosis and exocytosis, those are active transport. Those require energy. So C and what was that one? C and F. You'll go ahead and put those in because we haven't really gone over those yet. And then those same words are repeated again a little bit further down. So once again, let me see where they're at. G, where it says avacuole, that's G, and the one below it is F. So the other parts of it, so it's just using kind of your definitions, and then in the little box to start off with, and also the ones on the back, they're very similar to what we did yesterday. And then there's a couple of pictures. So that'll kind of summarize passive transport, and that's where we're going to finish up with our notes. So Cody, that's in Google Classroom under daily work. Did you find it?
I did it.
You already did it?
Yeah, I just did it.
Like you did it yesterday?
No, like I did a few minutes ago.
No, I'm asking you like you did the one yesterday, because you didn't really do the one yesterday.
There was one yesterday?
The ones with the percent, you only did half of it.
What? Yeah, I did the one from yesterday. Yeah.
Only did partial. That's what I'm asking. Okay. I see that you've completed this one. Alrighty. Then I guess we will see you on Monday for the journal test. Are you going to be back at school? Did you tell us, or am I confused?
I'm not going to be back next week. No, I'm in quarantine.
But you said you had your journal, right?
Yeah, I already have my journal. I'm good.
Okay. Alrighty, then we'll see you on Monday.